Hello, everyone. I want to welcome you back to these sessions. It's my privilege to be here today. My name is Sunny Coley. I am the head of medical and regulatory affairs at Roche Canada, and I'm a practicing physician in internal medicine and ICU at Oakville Hospital and adjunct faculty at McMaster. So it's a privilege for me to be involved in this today. I'm happy to join you as chair for the second portion of this event. At Roche, we are passionate about advancing science, no matter where it comes from. Only by partnering with the brightest minds can we serve patients with difficult to treat diseases and make sure that we deliver the right innovation to the right patient at the right time. And that mandate certainly resonates with me. In my previous role, I've been in this auditorium a lot. I was the co-founder of a digital health startup company that was actually supported by Mars. So I lived here and I lived in this auditorium, so it's weird to be here in another role. So innovation is a mandate it's really near and dear to my heart. So that is why I'm honored to introduce the leading researchers of our time who are pushing the boundaries of biomedical science. So with that in mind, our next early career investigator speaker is Dr. Anna Maria Copaescu. She is an allergy, immunology, and internal medicine specialist at the rank of assistant professor at McGill and the University Health Center in Montreal. She was selected by Dr. Deborah Cook as part of a team leading a multi-center randomized control trial on point-of-care penicillin allergy testing, which is near and dear to my heart as an ICU doctor and an internist. So I'd like to welcome Dr. Copaescu to the stage.
So thank you very much for that lovely introduction. I see people still coming in. I will wait just a couple of seconds, but I'm going to start. So my aim is really to convince you that by improving patient care, we can also focus on penicillin allergy. So first of all, I wanted to thank very much Dr. Cook for the opportunity to present today. I also wanted to thank the foundation very much. As you all know, Dr. Cook is a distinguished professor at the McMaster University in critical care medicine. By designing and conducting landmark national and international studies, her work has helped decrease the important human and economic costs of critical care illness. Her work has a direct impact on patients, their families, the healthcare systems, and societies. Her passion for creating measurable benefits for our patients is truly an inspiration for our particular research. So why is our research critical? It has been estimated that by 2050, 10 million worldwide deaths could result from antimicrobial resistance, making it deadlier than cancer. So let's try to understand how antibiotic resistance is developed and focus on antibiotic allergy and particularly penicillin allergy. Indeed, 10% of the Canadian population will report a penicillin allergy. However, it is now known that less than 1% are truly allergic. Please also note that when we focus on the severe cutaneous adverse drug reactions that have important mortality and morbidity, these have been reported in about 1 in 1,000 patients. So when a patient actually has a reported allergy or a true allergy to penicillin, this will lead to the prescription of broad-spectrum alternative antibiotics. The use of these antibiotics has been unfortunately associated with an increased healthcare cost, increased risk for antimicrobial resistance, suboptimal antibiotic therapy, medication error. Using these broad-spectrum antibiotics leads to undesirable side effects and also risk of hospital complications. It is needless to say that this antibiotic allergy is a national and international public health concern. And in this context, there has been increased focus on penicillin allergy and its impact on antimicrobial stewardship and antimicrobial resistance. Therefore, delabelling or the removal of the penicillin allergy or a drug allergy is now a known weapon in the fight against superbugs and antimicrobial resistance. I was fortunate enough to be actively involved in a multidisciplinary team involved in removing these allergy labeling, so the delabeling in hospitalized patients, and was able to witness the positive impacts on our cohort of patients. So let's now try to understand how a penicillin allergy label or a drug label has an impact on our patient's hospital stay. Here we have a patient that enters the hospital with a penicillin allergy, for example. During his hospital stay, this patient might unfortunately develop an infectious episode. Because he was prescribed broad-spectrum antibiotics, this person can have another unfortunate adverse drug reaction. When the patient leaves the hospital, he will have gained new adverse drug reactions. So what can we actually do when a patient reports a drug allergy? One of our clinical in vivo tools that we use on a daily basis on the allergy clinic is skin testing. Following negative skin tests, we understand that more than 85% of all penicillin allergy labels can be removed. Our gold standard in allergy, and drug allergy particularly, is drug ingestion challenge. And this test involves giving a test dose to our patients. If the patient develops no adverse drug reaction, we can then remove the allergy label, or what we just mentioned, the delabeling process. In our cohort of low-risk drug allergy patients in the hospital setting, we have managed to demonstrate that 96 to 99% of low-risk penicillin allergies can be removed by oral challenge. So are there actually any clinical scores that could help us risk stratify drug allergies? The PenFast tool is an internally and externally validated clinical decision rule. This tool has been developed by our group, led by my mentor, Dr. Trubiano, and the risk of a positive allergy test can be accurately predicted using this clinician decision rule. As you can see on the right side, with a score of less than 3, this is associated with 5% chance of developing an allergic reaction. And this actually brings us to our trial, the PALACE study, or the use of a penicillin allergy clinical decision rule to enable direct oral penicillin challenge. This is the first international multi-center non-inferiority randomized controlled trial. Our main question is, do we still need to perform skin testing for patients with low-risk penicillin allergies as defined by a PenFast score of less than 3? As I mentioned, this is an international trial. With our site in Canada, we are fortunate to collaborate with 2 centers in the United States. And finally, in Australia, where I participated in the design of the study during my fellowship, and I am very fortunate to currently be part of the leadership team. Our patients are currently being randomized to either skin testing, followed by oral challenge if skin testing is negative, or direct oral challenge. The randomization is currently stratified by clinical site, and the sample size that we aim is 380 patients. The recruitment is ongoing, and we have managed to recruit 351 patients today on both arms. So we consider that the ability to deliver point-of-care penicillin allergy investigations for this large cohort of patients without skin testing will improve patient access to testing and the use of preferred penicillin antibiotics. The data will be collected from all the centers that are implicated and will be analyzed together, and we hope that this will lead to local practice change, but also be considered as part of new drug allergy guidelines. When I am not focusing on delabeling the 10% and fighting antimicrobial resistance, I am very passionate about severe confirmed allergic reaction. My focus is to actually focus on the true allergic reactions and develop ex vivo immunological assays in the lab. The aim of these tools is to have no risk of drug re-exposure for our patients, as well as evaluate drug probability and guide safe drug reintroduction. In our work, we managed to demonstrate that a combination of in vivo, such as skin testing, and ex vivo diagnostic tools in the lab can help in the context of severe delayed immune-mediated antibiotic adverse drug reactions, can help improve causality assessment. I had the opportunity to study various broad-spectrum antibiotics, such as piperacillin, tazobactam, Bactam, amoxicillin, clavulanic acid, and currently Septra, in order to understand how we can improve ex vivo tools in the immunological lab. So based on the data from the PALACE trial, how can we improve hospital care and optimize care for our patients? How can we optimize the use of critical care medicine by improving antimicrobial stewardship? In an ideal hospital setting that you can see on the slide, the patient with an allergy label would be assessed by an inpatient surveillance program as soon as the patient is admitted to the hospital. This will lead to an antimicrobial stewardship evaluation and hopefully the removal of his drug allergy label before the patient actually developed his infectious episode. If the patient did end up developing, unfortunately, an adverse drug reaction following the prescription of his medication, this would be followed by another antimicrobial stewardship evaluation in order to guide appropriate use of antibiotics and appropriate use of medication for the rest of his stay and for future hospital stays. Unfortunately, if the patient will leave the hospital with persisting drug allergies, this will lead to an outpatient allergy evaluation where we would have the possibility to optimize our in vivo skin testing and ex vivo immunological assays in order to help predict what medication led to that adverse reaction. Over the following years, I really plan to understand translation and implementation research in drug allergy and how we can optimize drug allergy diagnostic tools. So as we discussed, allergy delabeling is a critical component of broader antibiotic stewardship interventions. I do hope I managed to convince you that removing a penicillin allergy offers the potential for enormous impact on both patient outcomes and healthcare costs. Defining safe and accessible pathways for penicillin allergy risk assessment and testing is essential to address this significant public health issue. Dr. Cook is an inspiration to researchers worldwide, as her studies have transformed critical care medicine and have led to an improved global healthcare that is focused on patients, practice, and policy. As penicillin allergy is a significant public health issue in a high-burden population, I am also optimistic that our research will have long-lasting impact on the management of drug allergy. And I would like to take the time to thank all the wonderful people that are making drug allergy research a better place. I am surrounded by amazing mentors and collaborators, both locally and internationally. Thank you very much for your attention.